ReactLab EQUILIBRIA is designed to analyze multi-wavelength spectroscopic measurements collected during automated or manual titration experiments. The aim is to quantitatively analyze the underlying reaction mechanisms to determine the equilibrium or formation constants for each step, as well as compute the spectra and concentration distributions of all the participating species. All data, reaction models and results are stored in pre-formatted Excel workbooks. ReactLab interacts with these workbooks to perform the analysis. I will now start ReactLab and load my example workbook containing measurements collected during a metal-ligand titration experiment. When a workbook is first opened, any data are automatically displayed in an interactive three-dimensional graph. The measurements themselves are stored in a specific format in the data worksheet of the workbook. The measurement spectra are organized in rows with the measurement wavelengths along the top. The titration additions are listed in a column to the left under the VAD heading. During analysis in automated titration mode, ReactLab will compute the total component concentrations in the measurement cell or beaker following each addition in order to calculate the concentration of all the species specified in the reaction mechanism. The columns to the right of the VAD vector will be used to hold these results. Alternatively, in manual mode, when a series of samples have been pre-prepared, these total component concentrations can be entered by the user. We are now ready to move to the main worksheet to enter our reaction model. The data we have provided is from a metal-ligand titration experiment. In this case, the metal can be doubly coordinated with two ligand molecules. Our aim is to determine the two corresponding formation constants. Whilst we have chosen to use formation constant notation, we could just as easily have written the model using equilibrium constants. In this case, the second step would be entered as ml plus L in equilibrium with ml2. ReactLab will automatically interpret the notation during model compilation. For further information on the use of these alternative formats, I refer you to the ReactLab EQUILIBRIA manual. The next step is to compile the model. To do this, we need to switch to the ReactLab GUI and press the Compile Model button. Once we have done this, we can return to the workbook. During compilation, ReactLab reads in and interprets the model, identifying all the interacting reaction components, M and L in this example, and all the reaction species according to the mechanism. These names are written back to the workbook to provide column headings for the model-dependent configuration settings and the output of analysis results later. First, we must define which species contribute spectrally to the measurement. In this example, we will define the metal and its complexes as coloured and the free ligand as colourless. We can also supply a known spectrum for any of the species. If so, these must be supplied in the AUX worksheet. Next, we add the component concentrations in the beaker and burette. These are required for the automatic calculation of the total component concentrations by ReactLab. In this experiment, we have 0.1 molar metal in the beaker and 0.5 molar ligand in the burette. We also enter the total volume in the beaker to start with, in this case 10 mils. Please note, if we leave the VTOT cell blank, this will signal to ReactLab that we require manual titration mode, and instead of calculating the total component concentrations and writing them back to the datasheet, ReactLab will read in values provided by the user. Next, we must enter values for the reaction parameters, including estimates of those we want to fit. For formation and equilibrium constants, we fit the logarithm of the actual parameter value. We select which we want to fit by ticking the appropriate checkbox. In this case, we're going to fit both formation constants. Now we can return to the ReactLab GUI to start the fit, but first we can use the update feature to validate our initial settings. This function computes how well the model with our initial parameters matches the data prior to undertaking the fit itself. We can see these starting parameters do not result in a good match of the model to the data from the distortion in the residual surface and the negative excursions of the spectra, but they will provide an adequate starting point for the fitting algorithm. If we now press the Fit button, ReactLab iteratively adjusts the parameters until the residual square sum converges to a minimum. The residual surface shows no systematic deviations and is uniformly random, and all the spectra are now positive. The best fit parameters with their standard errors and the final square sum are written back to the main worksheet. ReactLab has also populated the results sheet with the best fit concentration profiles and species spectra. It is now, of course, possible to use standard Excel tools to further manipulate the results or to create specific charts and graphs. This, of course, does not require ReactLab, and so can be carried out on any PC with Excel installed. The ReactLab workbook is therefore a complete and portable snapshot of the analysis session. We can also visualize the results in more detail from within ReactLab by launching the graphic GUI figure. Here we can see all the spectral measurements superimposed. I can overlay the calculated Y values from the model and can view the traces individually or scan through them continuously. I can switch the x-axis to observe the measurement profiles and residuals at specific wavelengths. We are also able to inspect the fitted concentration profiles and spectra of the species more closely in this figure. Any of these graphs may be directly printed out or saved to the Windows clipboard for insertion in other documents. To conclude, I will now return to the data sheet of the workbook to make some final remarks about total component concentrations, which in this example were calculated by ReactLab. The VTOT column has been calculated using information from the VAD column, and this, together with the initial concentration information in the main worksheet, has enabled the calculation of the concentration totals for the two components M and L. As was discussed earlier, by leaving VTOT blank in the main worksheet, the user can provide these values manually for pre-prepared samples. This must be done after model compilation, since ReactLab will determine the component names and provide these as the column headings. That concludes this demonstration. I can now save the workbook, which provides a complete snapshot of the analysis session. This can be reloaded into ReactLab later on to continue or revise the analysis, or can be reviewed and processed by Excel independently. Thank you very much for your attention.